Right now, if you are a man who practices intermittent fasting, your testosterone story is being written by a variable you almost certainly have never been told about, not by how many hours you fast, not by what you eat in your feeding window, not by whether you train, by a single biological reality about your starting point that determines whether the same fasting protocol that crashes testosterone in one man quietly rebuilds it in another, and the research that explains this is one of the most important, most misunderstood, and most dishonestly presented topics in all of men's health science. Here is the part that almost nobody in the fasting community addresses honestly. Every article about fasting and testosterone chooses a side. Either fasting destroys your hormones and you should never skip breakfast, or fasting is a testosterone supercharger and the gains are coming. Both of those framings are wrong. Both of them are serving an agenda, and the biology sitting between them is considerably more interesting, considerably more useful, and considerably more specific to the actual situation inside your body right now than either headline will ever tell you. This episode covers the complete testosterone picture of long-term intermittent fasting for men, the direct hormonal effects of fasting on the HPG axis, the hypothalamic pituitary gonadal axis that controls testosterone production from the brain down, the aromatase mechanism, the enzyme hiding inside your fat cells that is silently converting your testosterone into estrogen right now, regardless of whether you fast. The cortisol variable, the stress hormone that fasting can either suppress or amplify depending on protocol length, and why that single variable determines whether long-term fasting builds or degrades your endocrine system, and the body composition pivot, the point at which everything changes and the research stops being contradictory and starts telling a completely coherent story. By the end of this, you will know more about what fasting is doing to your testosterone than most men's health clinicians will ever tell you. Before the research makes complete sense, you need to understand the system it is operating on, because testosterone is not a single hormone sitting in isolation. It is the output of a communications chain that runs from your brain to your bloodstream, and every step in that chain is vulnerable to disruption. The hypothalamus, sitting at the base of your brain, releases gonadotropin-releasing hormone, GnRH, in rhythmic pulses. GnRH travels to the pituitary gland, which responds by releasing luteinizing hormone, LH. LH travels through the bloodstream to the Leydig cells in your testes, which convert cholesterol into testosterone. This is the HPG axis, your body's testosterone manufacturing line, and it runs on signals, not on calories, not directly on food timing, on signals. The question of what fasting does to testosterone is fundamentally a question about which signals fasting sends to each stage of that chain. And here is where the research produces its first important and frequently misrepresented finding. Short-term extended fasting, the kind involving 48 hours or more of complete food restriction, does send a suppressive signal to the HPG axis. A study published in the journal Endocrinology and Metabolism documented this precisely. Basal serum testosterone fell significantly in healthy men of normal weight after 48 hours of fasting, and luteinizing hormone fell alongside it. When the same researchers maintained blood glucose with small oral doses of glucose during the fast, the testosterone suppression did not occur. The mechanism behind the drop was not fasting itself, it was the glucose crash that accompanies it. The HPG axis reads falling glucose as a signal of genuine energy emergency, and under genuine energy emergency, testosterone production, which is metabolically expensive, is suppressed. The testes receive less LH signal, output drops. This is the finding that anti-fasting content loves to cite, and it would be definitive except for one critical detail. That study used 48-hour total fasting. You are almost certainly practicing 16/8 or something similar. Those are different physiological events. But here's the finding that most fasting advocates love to cite and that requires equal scrutiny. A systematic review and meta-analysis found that Ramadan fasting across 35 studies and 1,107 participants produced no significant changes in testosterone, LH, FSH, or prolactin. The HPG axis in the context of daily time-restricted eating, where calories are still consumed within a window, was not significantly disrupted. The system that regulates testosterone appeared largely stable. The honest synthesis is this: the effect of intermittent fasting on the HPG axis in men is protocol-dependent. Daily time-restricted eating at 16:8 or similar does not produce the acute hormonal suppression that multi-day total fasting does. The two are not equivalent physiological events and should never be cited as if they are. But here is where the fasting and testosterone story gets genuinely extraordinary, and where most of the health content you have ever consumed on this topic has failed you entirely. Because the direct hormonal effects of fasting on the HPG axis are half the story. The other half is happening in your fat cells right now, and it is operating completely independently of whether you fast. There's an enzyme called aromatase. It lives primarily in adipose tissue, particularly visceral fat, the deep abdominal fat surrounding your organs. Its biological function is to convert androgens into estrogens. Specifically, it converts testosterone to estradiol, the primary form of estrogen. And it does this continuously in proportion to your total visceral fat mass. The more visceral fat you carry, the more aromatase activity you have, the more of your circulating testosterone is being converted into estrogen before it can ever reach the receptor sites in your muscle, brain, or testes that need it. Research published in the Journal of Clinical Endocrinology and Metabolism confirmed that approximately 80% of circulating estradiol in men is produced through aromatization of testosterone in extragonadal tissues, including adipose tissue, and that aromatase gene and protein expression in subcutaneous fat was significantly higher in men with obesity compared to non-obese individuals, positively correlated with BMI, body fat percentage, and waist-to-hip ratio. This is not a marginal effect, it is a systemic hormonal recalibration driven entirely by body composition. The man with significant visceral fat is not just storing energy, he is running a continuous estrogen conversion factory that is actively depleting his testosterone supply. And here is where intermittent fasting's relationship to testosterone stops being confusing and starts being coherent. Because the most consistent and well-documented hormonal effect of long-term intermittent fasting in men is visceral fat reduction. A systematic review published in an umbrella analysis of randomized controlled trials confirmed that intermittent fasting significantly reduces fat mass and waist circumference. As visceral fat declines, aromatase activity declines with it. The conversion of testosterone to estrogen slows. Circulating testosterone levels rise, not because fasting directly stimulated the HPG axis, but because it dismantled the fat-based conversion system that had been silently draining testosterone for years. This is the aromatase pivot, and it is the most important variable in understanding what long-term fasting does to male testosterone that almost no health content will clearly explain. Think of testosterone as water flowing through a pipe. The HPG axis is the source, the tap. Aromatase is a leak in the pipe. Most conversations about testosterone focus entirely on the tap, trying to increase the output at the source. What fasting does long-term in men carrying excess visceral fat is repair the pipe. The output at the source remains roughly the same, but the leak stops. And the amount of testosterone arriving at its destination, your muscles, your brain, your testes, your well-being, increases substantially, not because the factory produced more, because less was being lost along the way. A review published in Juntendo University's medical journal captures this dynamic precisely. Even though acute fasting periods might decrease serum testosterone, the mechanism potentially involved in the regulation of testosterone through long-term fasting is the reduction in body fat and the following decrease in aromatase activity, reducing the conversion of testosterone into estradiol. In obese individuals, the LH to testosterone ratio, considered a marker for testicular function, significantly increased after sustained caloric restriction, pointing to improved testicular function alongside reduced aromatase activity. Now, let's talk about the cortisol variable, because this is the mechanism that determines whether your fasting protocol supports or undermines everything we just described, and it is the one most people practicing fasting get dangerously wrong. Cortisol is your primary stress hormone. It is released from your adrenal glands in response to perceived threat, whether that threat is physical, psychological, or metabolic. And it has a direct, well-documented suppressive relationship with testosterone. When cortisol is chronically elevated, it directly inhibits GnRH release from the hypothalamus, which reduces LH signaling to the testes, which reduces testosterone output. The mechanism is clean, linear, and dose-dependent. More chronic cortisol, less testosterone. This is the pathway through which chronic stress in any form, work, relationships, or dietary restriction, can systematically erode a man's hormonal health over months and years. Here's what the research shows about fasting and cortisol. The evidence is protocol-dependent and dose-dependent. Daily time-restricted eating in the 16 to 8 range conducted with adequate total caloric intake and sufficient protein does not produce significant chronic cortisol elevation in most healthy men. The adrenal signal fired during the fasting window is managed within normal physiological parameters. Extended fasting of 2 to 5 days or more, particularly with significant caloric deficit, does produce measurable cortisol elevation. And here is the finding that most people completely miss. Inadequate protein during the feeding window creates the same cortisol signal as undereating because your body cannot distinguish between strategic fasting and genuine food scarcity. If your feeding window is 8 hours, but you are consistently under-eating protein and total calories, your hypothalamic-pituitary-adrenal axis reads the same signal as starvation, and the testosterone consequences follow accordingly. This is why lean, physically active young men who fast aggressively with insufficient caloric intake show testosterone reductions in the research. A review of human trials published in Nutrients confirmed that intermittent fasting reduced testosterone in lean, physically active young males. These are men who do not have significant visceral fat to lose, meaning the aromatase pivot that benefits overweight men is not available to them. They get the direct HPG suppression from energy restriction without the compensatory benefit of reduced aromatase activity. For this population, aggressive fasting without adequate nutritional support is genuinely counterproductive for testosterone. But here is the complete, honest, and extraordinarily useful picture that emerges when all of this is assembled together. The effect of long-term intermittent fasting on testosterone in men is not a single outcome, it's a spectrum, and your position on that spectrum is determined almost entirely by your body composition at the point you begin. If you carry excess visceral fat and have the insulin resistance and elevated aromatase activity that accompanies it, long-term intermittent fasting conducted at 16 to 8 with adequate total calories and protein is likely to improve your testosterone profile over time. Not because fasting directly stimulates the HPG axis, but because it progressively dismantles the visceral fat and its embedded aromatase machinery, reducing estrogen conversion and allowing more of your available testosterone to reach the tissues that need it. If you are lean, active, and metabolically healthy, long-term intermittent fasting may have no significant effect on your testosterone, provided your feeding window contains sufficient calories and protein to meet your energy and muscle-building requirements. The risk profile is low, and the metabolic benefits remain real. If you are lean and under-eating, either because you are not attentive to your feeding window nutrition or because you are using fasting as a caloric restriction tool at the expense of adequate nutrient intake, long-term fasting carries a genuine risk of testosterone suppression through the cortisol pathway and through insufficient substrate for testosterone synthesis. 3 men, same fasting protocol. 3 completely different hormonal outcomes, determined not by the fasting itself, but by the biological reality each man brought to it. And here is the nuance that this research absolutely requires. The human trials on long-term intermittent fasting and testosterone are limited in number, variable in quality, and mostly short in duration. A 2024 systematic review by Kalsekar found mixed results across studies, with some showing significant reductions in total testosterone and some showing no significant change, and with the evidence base still too thin to draw definitive long-term conclusions. The aromatase mechanism is well-established physiology. The visceral fat and testosterone relationship is robustly documented. The cortisol suppression pathway is well understood, but the specific long-term hormonal trajectory of a man practicing 16 to 8 for 12 months, 2 years, 5 years, has not been comprehensively studied. The honest position is that the directional evidence is coherent and the mechanisms are solid, but the long-term clinical data is still being built. What this means practically is not uncertainty, it means precision. Your testosterone is answerable not through general advice, but through knowing your own body composition, your own caloric intake, your own cortisol load, and your own blood work. A testosterone panel before and 6 months into a fasting practice costs relatively little and provides more actionable information than any article, including this one, ever will. Here is the complete picture. Your testosterone is produced by a manufacturing chain running from hypothalamus to pituitary to testes. Short-term extended fasting suppresses that chain by dropping blood glucose below the threshold the HPG axis requires to function normally. Daily time-restricted eating does not produce this suppression in most men. Visceral fat contains aromatase, an enzyme that continuously converts testosterone to estrogen in proportion to fat mass. Long-term intermittent fasting reduces visceral fat, reduces aromatase activity, and allows more of your available testosterone to reach its destinations. Cortisol directly suppresses testosterone production, and inadequate caloric or protein intake during the feeding window triggers the same cortisol response as genuine starvation. The net outcome of long-term fasting on your testosterone depends on whether you are lean or carrying visceral fat, whether your feeding window is nutritionally sufficient, and whether your overall fasting protocol is creating a recoverable hormonal environment or a chronic stress one. Your body is not arbitrarily choosing between building you up and tearing you down. It is reading the signals you send it with extraordinary precision and responding to what those signals tell it about the environment you are living in. The aromatase factory in your visceral fat does not care about your intentions. It only cares about your biology. Give it less to work with through sustained visceral fat reduction from a well-executed fasting protocol with adequate protein, and it will give you more testosterone back. That's not a promise, it's a documented mechanism. The research has mapped it. The rest is execution. Drop a comment right now. Have you had your testosterone tested before and after starting a fasting practice, and what did the numbers actually show you? I read every single one. If this gave you the most honest and specific explanation of fasting and male hormones you have ever encountered, hit subscribe.
Every week in this series, I cover the fasting research completely, including the findings that complicate the simple story, with no products to sell and no supplement agenda to protect. And next, the complete hormonal optimization protocol for men who fast, the resistance training data, the protein timing research, the sleep quality variable, and the specific eating window strategy that protects the HPG axis while maximizing
every metabolic benefit fasting produces. Subscribe so you are there when it drops. The aromatase factory has been running without your knowledge. Now you know where the switch is.